Hi, welcome to your virtual class on photosynthesis. So let's warm up by thinking about cellular respiration. In cellular respiration, organic carbon-containing molecules, such as glucose, are transformed. So what happens? I have a little hint on this diagram here. We start with an organic molecule like glucose, and through the steps of cellular respiration, that carbon is taken off of the glucose in the form of carbon dioxide. Now let's compare that to photosynthesis overall. So in cellular respiration, we start with something like a 6-carbon sugar, and we're oxidizing it to CO2. Try to remember what the word oxidized means. It's talking about the transfer of electrons, and oxidation is when something is losing electrons. So glucose loses electrons throughout the process, and we end up with CO2. In the case of photosynthesis, it's the reverse. We're going to be using carbon dioxide from the atmosphere. The plant's going to be using carbon dioxide, and it's going to be fixing that carbon dioxide to build organic molecules like sugar. So, photosynthesis uses solar energy to produce sugar from carbon dioxide and water. If we think about a plant, we know that there's photosynthetic leaves, that underground we will have roots. Okay, so for a plant, they breathe in carbon dioxide, and the gases that they breathe out through their leaves is oxygen. This is a part of photosynthesis, and it's really beautiful because it is the opposite of what humans do. So we complement each other in that way, and we can only survive because of this activity of plants. We also know that there's 2 other really crucial key components of photosynthesis. There has to be an energy source. So light is captured by chlorophyll in the plant leaves, and we also need water. We know that water is taken up through the roots and brought up to the leaves. So try to pause and remember the term. What do you call an organism that gets its energy from sunlight? I know that you really want to say autotroph, but it's not the answer here. A phototroph is an organism that gets its energy from light in the form of photons. Okay, so then what does autotroph mean? Well, it actually has to do with carbon source. Autotrophs are sometimes called self-feeders, but again, it's not really because of the sunlight. It's the fact that instead of eating other organisms to obtain carbon in the way that we do, they get their carbon from an inorganic source, carbon dioxide. Here's a handy table to kind of break down these differences in metabolism. We can have carbon sources that are organic, and in that case, organisms are called heterotrophs. If the carbon source is inorganic, then the organism is called an autotroph, like in the case of plants. Then we can also be thinking about where energy is coming from. If an organism gets its energy from light, it is a phototroph. If an organism gets its energy from breaking chemical bonds, it's a chemotroph. So humans, we get our energy from breaking chemical bonds that are found in organic molecules like glucose. So we are chemotrophs. We also get our carbon from that same source, and that's an organic source. So we are chemoheterotrophs. Plants, on the other hand, are photoautotrophs because they get their energy from light and their carbon from an inorganic source, CO2. There are organisms, especially in the microbial world, that can fit into each of these categories. But for today, when we're talking about photosynthesis, we're going to be very eukaryote-centric. We're going to be focusing on the eukaryotes and mostly plants. But just remember that in the microbial world, there's a ton of diversity out there when it comes to metabolism and how organisms get their energy. So in eukaryotes like plants, where does photosynthesis take place? Well, we have two main reactions, and usually it's split up by Act 1 and Act 2. Act 1 being the light reactions. Generally speaking, everything's happening in the chloroplast, but the light reactions happen in the thylakoid membranes. So if you check out this image, you see these pancakes. They kind of look like pancake stacks. Those are our thylakoids. And there's the lumen of the thylakoid, which is, lumen means interior, inside, and then membrane of the thylakoid. So they are also membrane bound, also made up of phospholipids like typical membranes that surround organelles or the cell membrane. In the light reaction, we're going to be capturing light energy and converting it to chemical energy. Remember, first law of thermodynamics, we can't create energy from nowhere. It's always energy transfers. So in this case, we're going from light to chemical energy. And that's really the amazing thing about plants, is that they have the ability to do this. Second act is called the Calvin cycle. This is happening in the stroma, which is outside of the thylakoids, but we are still in the chloroplast. And this is part 2. So this is when we use electron carriers plus ATP that was all generated in that first step to make sugars. So think like step 1 is I'm capturing light energy, I'm transferring that energy. Step 2 is I'm using the products I made from step 1 to actually build sugars that I can store in the cells for later. Here's a full breakdown of the process. If I were to kind of split it up by part 1 and part 2, it would be like a line going down the middle here. On the left side is part 1. You see light coming in for the light reactions. It's hitting, our little pancake stack in the chloroplast. On the top of this image, we see things like water and carbon dioxide. These are going to be our inputs or our overall reactants. So water and sunlight are reactants for those first light reactions. And oxygen is actually a byproduct. You'll also notice here that ATP, so think like energy currency of the cell. It's kind of like money. It's little energy-packed molecules that you can spend around the cell whenever you'd like at any time. A little ATP is made. It's going to fuel the Calvin cycle, which is part 2. And we also had our electron taxis, aka electron carriers, get filled up with some electrons in part 1 in the form, and now we have NADPH. It's very similar to NADH that we learned about in cellular respiration. In the Calvin cycle, we're using products from part 1 and taking in carbon dioxide as our carbon source to build sugars. So remember, building pathways require energy, and that's why in part 1 we made ATP and we also harvested some electrons so that we can build those sugars. So let's get into part 1, or Act 1, the light-dependent reactions. Okay, what is light energy? This is kind of an abstract concept, I'm gonna have to admit it, but light energy is electromagnetic energy. It's composed of photon particles. They are both an energy packet and a wavelength at the same time. It's crazy physics stuff. And visible light, what we can see as humans, is just a small part of this spectrum, but turns out that plants also use the visible light spectrum. So coming back to photons, think about them as these little packets of energy, and they have 2 fates. Okay, so when photons are coming down from the sun and hitting a plant leaf and hitting the chloroplast, there's 2 possible things that can happen. The photons can be reflected or transmitted, or the photon can be absorbed and that energy is converted to another form. So you can guess what's happening in photosynthesis. That light is being absorbed. And we all know, if you take a look around, unless it's fall time, the plants around you are typically very green. So what's happening there? When we look at an object and it's green, like a plant, is the green light being reflected or absorbed? Think about it for a second. The answer would be that it's being reflected. It is not being absorbed by the plant. So plants have various pigments, and different pigments absorb different parts of the electromagnetic spectrum of visible light. So here we see chlorophyll A in part A, chlorophyll B, and then beta carotene as C. And you can see their chemical structures, and then right next to it in the part of this graph labeled D, circle it here, we see the absorption spectra of these chloroplast pigments. So if you take a look at these different peaks, you'll notice that there's higher absorption of light for purples, blues, tail end of blue, and then again towards the yellows and oranges and reds, but not much going on in that green area. And that's why green is getting reflected back at us, and all these plants appear as green. So we have these three pigments, and there can be various others that are embedded in the thylakoid membranes within the chloroplasts. This also helps explain why in the fall the leaves look so beautiful. What's happening is the leaves are actually going through senescence. They're starting to die, and they stop producing certain pigments. So colors that usually would be getting absorbed are now being reflected back at us, and we see different yellows and reds throughout the process of the plants shedding their leaves. So it's all thanks to pigments. Okay, let's check out the components of the thylakoid membranes. So we have what are called photosystems. I'll highlight them here with my pointer. There's two photosystems, and if we're going sequentially through this whole system here, it starts with photosystem 2 and then goes to photosystem 1, which feels like doesn't make any sense, and true. But the thing is, photosystem 1 was discovered first by researchers, so they named it photosystem 1, and then later it was discovered that there's also a photosystem 2. And it happens earlier in this whole process. So it's a little backwards, but that's just the way it is, and so we work with what we've got here. So we have these photosystems, and you'll notice that one of their jobs is in what we talked about with capturing light and converting light energy. So that's like big job of the photosystems. It also ends up exciting electrons. Okay, we also have electron transport chains, and there's two. There's an electron transport chain that happens between the two photosystems, and then there's a final electron transport chain where NADP plus gets reduced. Think about that for a second. Reduction is gaining, gets reduced to NADPH. We also have in this membrane one of our enzyme complexes, ATP synthase. This should look familiar if you've gone through cellular respiration. And NADP reductase, which we just talked about, where NADP plus gets reduced. We'll look at this in more detail, step by step. So first, let's take a closer look at our photosystems. So energy passes from sunlight all the way to the reaction center. So here's a photon hitting this protein complex, and you see these arrows moving through these little green circles. These green circles are supposed to be representing different pigments like chlorophylls, and the white arrows are showing the transfer of energy. Note that it's not yet transfer of electrons, just transfer of energy, and that energy is being moved through this protein complex, through the pigments, to arrive at this special pair of chlorophyll A molecules in the reaction center, it's called. So we have two of these chlorophyll A's. The light, that energy that has been transferred, excites electrons from the chlorophyll A pair and passes it to the first electron acceptor in the electron transport chain. So we initiate our electron transport chain. This makes sense. Electron transport chains, think about the name, they transfer electrons through usually a series of proteins embedded in a membrane. So where did the electrons come from? Here's where we answer that question. The electrons came from chlorophyll A, and we were able to excite those electrons from energy coming from the sunlight. So note, chlorophyll A, it lost electrons. If you think of the oil rig acronym, oxidation is losing. Chlorophyll A was oxidized. Now, it would be problematic if we just left it there because chlorophyll just lost its electrons. It's pretty much it won't be able to keep going with this process unless we have a way to replenish enzymes. So, electrons. So what's really cool here is we replenish the electrons that were lost from chlorophyll A with water. So water will give away 2 electrons and let go of 2 hydrogen ions. So remember that a hydrogen is just a proton and 1 electron. So it's often a way that we see electrons being moved around. I can take the electron off of it, end up with just hydrogen ions, the positive charge, just the protons, and these electrons are going off on their own journey. You'll also notice, this might look familiar, so in cellular respiration at the end of the electron transport chain, oxygen gas is the final electron acceptor, but it can't just pick up these electrons on its own, so it pairs up with hydrogen ions and we make water. Right here, we are seeing the literal exact opposite. Water is liberating electrons and hydrogen ions versus what we see in cellular respiration, which is accepting electrons. So the electrons get replenished through water. And that's where water comes in as super important in this process. So think about it for a second. Is water being oxidized or reduced? I always have to go through it in my head. Okay, water is losing electrons, oxidation is losing, so it's being oxidized. Okay, now we have an electron transport chain between the 2 photosystems. We just learned that light energy is coming in, it's exciting the chlorophylls, they pop off 2 electrons. Now it's going to go through this electron transport chain. So that's this first one that goes between the 2 photosystems. Similar to in what we saw with cellular respiration, these cytochrome complexes, these proteins embedded in that membrane, they have the ability to pass along electrons, right, and harness the little energy bursts that come with doing so. They're also transport proteins. So again, similar to cellular respiration, we're going to be building a hydrogen ion concentration. This time though, the hydrogen ion concentration is higher in the inner thylakoid space. So this is a little bit reversed if we're thinking of what happens in the mitochondria. Just note that. But the concept is the same. We're doing active transport and bringing hydrogen ions into the thylakoid space so that they can do the exact same thing that we see happen in cellular respiration. Those hydrogen ions, like a river flowing downhill, they want to go to where they're less concentrated. So they flow through a handy little channel, ATP synthase, and as they flow downhill, ATP synthase harnesses that kinetic energy and uses it to build ATP molecules. I really love thinking about this as like hydroelectric power, because with hydroelectric power, we have water flowing downhill, we stick a turbine in it, harness that kinetic energy and power light bulbs in our houses. It's basically that. The hydrogen ions are a river flowing downstream. We stick a turbine in there through ATP synthase, and that kinetic energy can be used to build ATP molecules. So we are almost ready for our second photosystem. Or to move on to part 2, but let's look at the photosystem 1, but the second one we're looking at. So we did photosystem 2. We went through the electron transport chain. We built a hydrogen ion gradient. We made ATP, but the show must go on. So those electrons that were moving from photosystem 2 to photosystem 1 arrive at photosystem 1. A similar process happens where we have light energy coming in, and that light energy gets passed around through the pigments and excites the electrons in those 2 special chlorophyll A's. The electrons are coming from the electron transport chain, or being replenished through this chain. So then those electrons are going to go through the final electron transport chain here. And you'll notice that they have to get picked up, right? They got to get picked up somewhere. And what happens is we have NADP plus reductase, an enzyme that helps facilitate the final transfer of the electrons. So we have NADP plus, which is just like NAD plus in cellular respiration that we learned about. It's an empty taxi. And it grabs onto those electrons and we have a full taxi, NADPH, that's ready to go drive somewhere else and drop off those electrons where they're needed. Now we're ready for part 2, the Calvin cycle. So a lot just happened here. I want us to think about what we just left with. So we had light coming in, we used water, created oxygen byproduct, we made some ATP, right, from kind of a part 1, moving from photosystem 2 to photosystem 1. We created that hydrogen ion gradient that allowed us to make some ATP. We also filled up our electron taxis and we have NADPH. So both of these things, NADPH, full electron taxis, and ATP are moving to the Calvin cycle, and that is Act 2. Before we dive in, let's go back to the big picture here. So what happened to our electrons? You can pause, think about where they came from. Hopefully you paused, but I'll tell you that they got excited off of those chlorophyll A's that are in the reaction center in the thylakoid, and they get replenished with water. Electrons coming from water molecules. The electrons went through an electron transport chain and eventually end up in our electron taxi, NADPH. Right, started on water, moved to NADP+, and become NADPH. Are we making or are we using ATP? In the process as a whole, both. But in part 1, Act 1, the light-dependent reaction, we are making ATP. In part 2, the Calvin cycle, we use that ATP, but the parts can get recycled. When we use ATP, we do ATP hydrolysis. It turns into ADP, adenosine diphosphate, and 1-phosphate group. Well, we can just take those parts and stick them back together. So we will take those parts and recreate ATP in the light-dependent reactions, which is why this is shown as a cycle. And then where we're headed is using carbon dioxide from the atmosphere as a carbon source to go through a series of chemical reactions that use ATP and our electron carriers and eventually build sugars. So let's talk about it, the light-independent reactions. Okay, in the light-independent reactions, first off, it's named that because it's independent of light, right? The harnessing of light energy all happens in part 1. Now we're using the products we created to build sugars. So CO2 is what enters this cycle. This is our main player. This is our carbon source. This is also why, to combat climate change, which is fueled by a rise in CO2 levels in the atmosphere, we can plant trees. We can plant a lot of plants that help grab carbon dioxide from the atmosphere and fix it into sugars, right? It's like, where does that carbon dioxide go when they breathe it in? They literally turn it into sugars, these plants. Okay, so there's generally like 3 stages to this Calvin cycle. The first would be called fixation. This is when that CO2 is added to RUBP by an enzyme called rubisco. Maybe for our sake, we don't need to memorize every single step, but essentially what's happening here is carbon dioxide is coming through. There's an enzyme that catalyzes a reaction to help build a larger carbon-containing molecule. Because we're building, ATP is used, right? And we also need some electrons. So that's where our electron taxis come in and they drop off the electrons that we harvested earlier. Now we get our G3P molecule. What's so special about it is G3P is something that we need to continue this cycle, to keep it going and fixing more carbon dioxide. And it's also the precursor to make things like glucose and other organic compounds. So the Calvin cycle is very different from the citric acid cycle, but something that I want you to see in terms of similarities is that we have an input molecule, and this metabolic pathway cycles around endlessly as long as we have inputs and outputs. So the plant will constantly take in carbon dioxide and it will go through a series of reactions that gives us something like G3P. which will continue to react with carbon dioxide and over again. So just to clarify, G3P is what we use to make sugars. To kind of give us some well-roundedness with this here, I do want to point out that plants are literally breathing in a similar way that humans are. Obviously, they don't have lungs like we do, but when we breathe in oxygen and breathe out carbon dioxide, we've learned that this is because of cellular respiration. In order to extract energy from chemical bonds in food, aka in order for me to get energy from the food that I eat for my breakfast every morning, I have to breathe in oxygen because it has a really important role of grabbing onto electrons at the end of the electron transport chain. And throughout cellular respiration in all of the cells in my body, I'm generating a bunch of CO2 and I need to get rid of that so it doesn't build up in my body. So I'm breathing it out. What's really cool is plants are the opposite, right? We just learned that they breathe in carbon dioxide. And as a byproduct of photosynthesis, they make oxygen. They breathe out oxygen. They are literally our complement. This beautiful evolutionary, journey together. Anyways, so plants are breathing like we do. They don't have lungs, but they do have basically like these little tiny mouths that are called stomata. So if you think of it as like a little mouth, I'll highlight this is the upper lip and the bottom lip. They're shown closed here, but they can open and close to allow for gas exchange or shut down gas exchange. Here's on the bottom, a cross section, and you can see that there's these gaps, and the stomata can actually like fill with water and pressurize or depressurize, allowing them to open and close. So plants really do coordinate their breathing and gas exchange. I bring this up because now we can kind of understand some real ways to apply this in terms of evolution and adaptations. So if you think about some plants that maybe live in, got cut off, but hot, dry environments, they're going to face some challenges when they're breathing. If I were you, I know nobody pauses the video, but if I were you, pause the video and think about it. What challenges is a plant going to face? They're trying to do photosynthesis. They have to breathe gases, but they live in a really hot, dry environment. Well, one of the issues is they open up their stomata to breathe. They are doing gas exchange, but they're also losing water because water evaporates very quickly in very hot, dry environments. So in those environments, there's also usually a scarcity of water. And these plants don't want to be unnecessarily losing precious water. I just wanted to bring up CAM photosynthesis because it is super cool and it's an example of an adaptation that's come about through long evolutionary processes to get around an issue like this. So during CAM photosynthesis, plants will, like a cactus, might open its stomata at nighttime. That way, it can fix carbon dioxide when it's at less risk of losing water from having its stomata open and doing gas exchange and breathing. It's going to store it as a compound called malic acid. Then, during the daytime, instead of opening those stomata to take in carbon dioxide, we'll use malic acid as like a store of carbon and use that to fuel the Calvin cycle during the day. So just a cool example of how plants, just one of many, many ways that plants are adapted to deal with different challenges they face. And now that you know the basics of photosynthesis, you can expand on it and learn about different evolutionary processes like this one.